In chapter 22, we really want to focus and follow what is happening with nitrogen in the body. In order to really understand nitrogen metabolism, we need to look at some of the processing of amino acids. We've previously looked at breakdown of amino acids, so now we want to look at what we can do to build the amino acids. One thing you should notice is that a lot of the reactions involved in the breakdown of amino acids are actually also involved in the production of amino acids, because what we're doing is following the movement of nitrogen. To get a true understanding of this nitrogen metabolism we'll study in this chapter, it is important that you go back and refresh your memory on the urea cycle, as some of that will tend to pop back up as we go through. I'm going to break this into 3 different parts. So first, let's take a look at the biosynthesis of amino acids, and then I'll record a couple follow-up lectures where we will then look at the way nitrogen is handled as we incorporate into nucleotides, both purine and pyrimidine. So starting, we want to take a look at amino acids. So first, let's think about what our body is going to do with amino acids. We really have 2 sources of amino acids for metabolic processes. We can think about our dietary proteins. Dietary proteins are going to be digested within the digestive tract and then transported into the blood in the form of amino acids. There are often times as well when endogenous proteins will be broken down into individual amino acids to place into the bloodstream. So regardless of what the source, we want to think about what's going to happen to the amino acids in our blood. Well, one thing we'll do with amino acids that we want to look at in this chapter is we can take these amino acids and we can produce proteins. Now, we're actually not going to go all the way to production of proteins, but we're going to look at building specific amino acids that we need so they can further be incorporated into proteins. Later in the chapter, we want to look at how our levels of amino acids in the blood can be incorporated into our nucleotides, our purines, and our pyrimidines. But you'll see from this list, there's quite a few other things that the amino acids will be used for. What we've studied previously is what our body does when we have sort of excess amino acids in the blood that we do not need to preserve the nitrogen. So we'll take those amino acids, remove the nitrogen, excreting through the urea cycle, and then use the corresponding keto acids that can feed into our other metabolic pathways such as glycolysis and the TCA cycle. So our first few reactions we want to take a look at should be familiar, as we looked at these when we studied the breakdown of amino acids. And our first reaction is using the enzyme aminotransferase. So you should recall that there are multiple aminotransferases depending on which amino acid. So it's named for the amino acid in which we're removing that amine group. So in this particular example, this is a transfer of the amine group from some amino acid to alpha-ketoglutarate, shown here. When we do this transfer, the alpha-ketoglutarate will receive the amine group becoming glutamate, and the particular amino acid in which the donation was made from will become its corresponding alpha-keto acid. So make sure you can recall this type of reaction. The second reaction that we did mention probably a little more briefly in our discussions was the glutamine synthetase. Now this is an ATP requiring reaction in which we will phosphorylate a glutamate, providing a good leaving group so that we can then add a free amine to produce glutamine. So recall from our previous lectures that we use glutamate and glutamine quite a bit. The major role for glutamate is sort of a storage site for our amines, because glutamate can transfer the amine to other keto acids, or glutamate can transfer its amine to the urea cycle for nitrogen excretion. Now flipping to the next slide, you also see this glutamine, very important. Recall that glutamine, along with alanine, were our main amino acids that provided transport of amine groups through the body, ultimately to the liver for processing. Since glutamate and glutamine are so important in the processing of amino acids and the transport of our amine groups, this is going to serve as a really important control point. So you can see that we have multiple amino acids that will inhibit the transfer of the amine group from glutamate to glutamine. This allows our body to determine we have a lot of amino acids available. So if we have a lot of amino acids available, let's go back and inhibit the production of glutamine. If we leave the amine group in the form of glutamate, that's going to trigger an increase in the urea cycle and further excretion of amine groups. Also, we see that AMP and CTP, some nucleotides, inhibit this incorporation of amine groups into glutamine. And we're going to see that this is related to, as we go into the end of this chapter, glutamate and glutamine are very important in the transfer of amine groups to produce nucleotides. So again, if we have a lot of product, we want to go back and inhibit some of those preliminary steps. As we look at the reactions that produce amino acids, we're going to see three common reactions that as you go through, I want you to pay special attention to these types of reactions. The first reaction is the first one we went over, the transamination reactions. You're going to see with this the use of pyridoxal phosphate, abbreviated PLP. This is actually a form of vitamin B6. So this is going to be this type of reaction that we just reviewed. The second reaction is the transfer of one carbon groups using a tetrahydrofolate derivative. And we're going to see this anytime we're donating or moving one carbon units around. And then the last type we'll see is transfer of amine groups that are derived from glutamine. So remember in class I told you this should be glutamine. So these glutamine aminotransferases. Since we see the glutamine aminotransferase so much, let's go ahead and actually look at the catalytic mechanism briefly. So if we look here at the catalytic mechanism, we see there's an important active site amino acid, cysteine. This cysteine is going to serve as a nucleophile, helping to break this amide bond in our glutamine. So glutamine will come in, bind to the binding site for glutamine. The cysteine residue helps with the reaction, breaking the bond to release the amine group. Now down here in the second part of the picture, we have the other binding site where whatever we are transferring the amine group is going to bind. The amine group can then bind to our substrate. We will release the aminated substrate molecule as a product, and we release glutamate, formed because the glutamine originally here lost its amine group. As we go through the different amino acid synthesis slides, you're going to see some repeat information, trying to give you as many visual different clues and ways to learn what's important. What I want you to focus on as you learn this amino acid biosynthesis is really what are your precursor molecules and what are some important steps as we go along. This slide here gives you a lot of the information that you'll need. So you see these that are shown here in kind of this orangey pink color. It shows you that some of the intermediates of glycolysis can either directly or indirectly produce some of our amino acids. Also shown here in the purple color, we can have intermediates from the pentose phosphate pathway leading to the production of amino acids, as well as shown here in light blue, intermediates of the TCA cycle can be used to directly or indirectly produce some of our amino acids. I've also provided for you this table from your textbook. It shows you all of the amino acids with the star. They are going to be what we consider essential amino acids, meaning as humans we don't make them. So don't get tricked up. Some of the pictures from your textbook are going to have some amino acids and reactions that we can't make as humans. So always refer back to this table showing you which amino acids we can make without a star. So now flipping through, this is what I want you to pay attention to. You should be able to visualize that a transamination from alpha-ketoglutarate can directly give you glutamate. Of course, you should recognize that glutamate can become glutamine. This is one of our big reactions we just reviewed. Arginine is another molecule that you should be able to remember we can produce, shown here, as part of our urea cycle. From 3-phosphoglycerate, we can produce serine. Don't memorize all the reactions, but the molecule itself is similar enough, you should see the relationship. And then from serine to glycine, this is simply an addition, or actually it's a removal of one carbon from serine to glycine. So you see that folate here becoming important. To produce cysteine, we'll take a serine that we made, combine it with a molecule that we obtain from methionine, several reactions, and we can produce cysteine. Getting more complicated, so we're going to look at our nice little abbreviated flowcharts we have. Oxaloacetate can amidate to produce aspartate. Aspartate can then produce asparagine. Pyruvate can be amidated to produce alanine. So these obvious ones you need to recognize. Very important in some metabolic disorders. We can see production of phenylalanine and then subsequently tyrosine. There's lots of issues with enzymes that produce phenylalanine leading to PKU. And then this histidine is one of our molecules from our pentose phosphate pathway. Ribose 5-phosphate is going to produce histidine. So this last slide that shows you just another view of how we can produce these amino acids. So it's, again, really important for you to follow what particular main pathway we've studied that's going to lead to the production of our common amino acids. And then, of course, don't forget, when we make amino acids, we're not just thinking about making the amino acid for the protein, or even just to produce the nucleotide. There are many other molecules that are derived from amino acids. And so this slide just shows you a few here that are essential to the human body.